next part of my lecture. So let's go look at this article. Let's see. I think it's this one. Agrobacterium, TI plasmid. Yeah, that's coming later. That's the immunology thing. So this is going to be the article. And boom, look at figure 1 there. You recognize that? Figure A, B, C, D, E, G. Yeah, you recognize that. You've got your auxins, cytokinins, and opines, and you're going to have, there are actually different versions of the TI plasmid that you can find in nature, and they will contain some of these proteins, not necessarily all of them. There are also some other things here that would sort of help the process go, and down here is the origin of replication. So that is a typical type of TI plasmid. There we go. Okay, down here, figure 2, easy. So this is going to show you that, yeah, let's see, we've got different, okay, so this is a generalized model of how these genes are going to be regulated. So there's going to be proteins that come out that then inhibit the production of other proteins. So you've got, proteins that drive replication and other proteins that are going to shut down replication. And so that's located elsewhere on the protein. That's not as important. Here is our, type 4 secretion system. And so there you go. You got, this is your little syringe. And these are the barrel, basically, of this protein. And so the idea is that it's going to insert the syringe like this, and then the syringe is going to change conformation, if I remember correctly. And so now it's going to look like that, and it's going to have a nice hole in the middle, and that's how we are going to inject that tDNA. It's going to go right through there. And so, yeah, then it'll be inside the cell, the plant cell. And here we go. These are some organization of some TI plasmids that have been found in Agrobacterium. You'll see a couple of these things that you recognize. Let's see. They would normally have their names above them. And so they're going to get called different names, but they're going to have the same functions as normal, I think. Okay, this is talking about the tDNA being transferred, and so it's going to go through the way some of those proteins are actually going to go about chopping out the DNA. And these are the things that Agrobacterium wants from the plant, these sort of sugars and nutrients. And so it's going to send that transfer DNA, that tDNA, in to actually become part of the cell and drive the production of auxins and cytokinins and opines, which cause tumors, basically uncontrolled cell proliferation. Okay, and that's the end of it. So this is probably more information than you ever wanted or needed, but it's explaining everything else that's going on around these particular genes. So this is the super advanced version, and this is where, I'd like you to be able to take at least something from a paper like this and add it into your answer, and that is then going to be a very good answer. Now remember, our tutors are open for business. They figured out tutoring via Snapchat and via Instagram. They got ways to help you as long as you have some kind of phone or internet connectivity. Just go online to the Student Success Center, just like you always did, or give them a call. There's still people there to answer the phones, I believe. I think they have the calls forwarding to them. So yeah, give it a try.